Good morning. I'm back with you all to continue the last topic of material referred to as cell reproduction and inheritance. We're going to talk about meiosis and its importance in sexual reproduction in this unit of chapter 11. It goes well with chapter 10 on mitosis as two cell reproduction types in eukaryotic cells. So let's start out with asexual versus sexual reproduction. Asexual reproduction is considered to be the primitive or simplistic mode of producing offspring. The offspring should be genetically identical to the parent if it reproduces asexually. Well, we refer to these as clones. It's fairly energy efficient from a standpoint of using energy, ATP, to do this asexual reproduction is fairly energy inexpensive, so it doesn't take a lot of energy. It can produce lots of offspring. Only drawback might be that the offspring are genetically identical to the parent. Again, there's not a lot of genetic diversity of genes, alleles in a population that produces asexually. That's not necessarily a problem, and if the environment is fairly stable, it can be actually the most efficient means of reproduction. Sexual reproduction, and most people consider the evolution of sex to be a kind of a quagmire, just because it takes longer. It involves, for animals, it involves elaborate male-to-male competitions or female choice. There's a lot of work involved. There's a lot of energy expenditure, even by plants producing flowers, attracting pollinators to flowers, then producing fruits, and then having to disperse seed, maybe by an animal, eating a fruit. So sexual reproduction has always been considered, well, why? Because of the energy expenditure and the time involved. And the main reason that people who have studied this topic have come up with is variation. Different genes, different alleles, a better means of adapting and changing to one's environment. And sexual reproduction enables a population to be genetically variable, and maybe a environmental change occurs and some members of that population can survive, whereas in an asexual population, maybe all members might perish. So take that for what it's worth. But sexual reproduction is certainly a very important means of producing offspring because of the spread of adaptations through the population. The main means of producing reproductive cells for sexual reproduction is a type of division referred to as meiosis. Meiosis, again, refers to the division of a nucleus of a cell, and this type of process will result in two divisions of the nucleus to form four haploid daughter cells. As is shown in the illustration, each of the daughter cells will characteristically contain different sets of alleles and be genetically distinct from one another and from the parent. So, meiosis forms haploid cells, gametes or spores. These gametes or spores tend to be genetically variable, and they spread variation within a population. In both animals and plants, the products that are male from meiosis all tend to survive. In animals, the male products are called sperm, and in plants, they're called microspores, male spores. They're called micro just because they're much smaller than the female spore, the megaspore, if you will. In animals and plants, only one of the female products of meiosis tends to survive to become a functional egg or ovum. Well, we refer to the female gamete as an egg or an ovum. We refer to the plant gamete to be a mega or a macro spore. So the female product, one survives to become a functional egg or ovum, and three disintegrate or degenerate. In animals, they're called polar bodies, if you will. So the male product all survive to be either sperm or microspores. In most plants, not all, but in most plants, the spores are produced within the flower, within specific structures of the flower. The male spores tend to be produced within an anther, and the female spores tend to be produced within an ovary. Once the male spores are carried via pollen to the female, most all parts of a flower will disintegrate. The ovary of a flower, if the eggs or ovules are fertilized, the ovary will swell up, mature as a fruit, and the ovules will mature as seeds. Within the human, testes contain the structure to produce male sperm, and females have two ovaries that produce eggs or ova. In both cases with the human, the reproductive system is connected to the urinary system, the urine, and is often referred to as a urogenital system. You'll review this when you go over the rat dissection lab and look at some of the illustrations and not live, but preserved models of the rat. You'll notice the connection between the kidneys and the reproductive structures in both male and female rats. And it's basically about the same in the human as well. So, meiosis is not one division of the cell, but two. We refer to these as meiosis 1 and meiosis 2. This slide gives an overview of the main events in each. The main processes of meiosis that make it distinct from mitosis occur in meiosis 1, and I have them listed in 2 bullets. Number 1, crossing over occurs, and that's going to be the last slide coming up next to talk about crossing over. And number 2, the number of chromosomes is reduced by 1/2. So at the end of meiosis 1, you have 2 cells formed that are genetically different, and they contain half the number of chromosomes as the initial parent cell. Meiosis 2, nothing really unique happens in that division. You start with 2 cells that are haploid, and you end with 4 cells that are haploid. The events in each of the stages are pretty much the same as what occurs in mitosis. So the main things that happen in meiosis to make it a distinct type of division occur in the first division called meiosis 1. I said we were going to talk about crossing over. Crossing over is really the means by which genetic variation is introduced into a population. At least it's one means. Another means of introducing genetic variation could be mutation. And there's others. Crossing over. So let's talk about it. It occurs in the first step of meiosis, the division of the nucleus of a reproductive cell, a germ cell. It occurs when a pair of homologous chromosomes, two, will fuse. That is referred to as synapsis. Synapsis, or fusion of homologous chromosomes, forms a tetrad. A tetrad is four. So that refers to as four chromatids being fused together. The regions where the fusion or the overlapping of the chromatids occurs is referred to as chiasmata. And those are regions where actually portions of the chromatids can break off and reattach. So look at the illustration. You have a purple and you have a blue chromosome. They're both duplicated and they kind of look like, to me anyway, the letter X. They fuse in letter C. Close contact results in fusion and there is a crossover event. So a chiasmata occurs, and you notice in letter D that the purple chromosome, the lower right chromatid, has a light blue piece attached to it. The light blue chromosome has a small portion of the purple chromosome attached to it. So a crossover event is shown. Part A shows the duplication of the chromosomes to form two chromatids, and then parts B, C, and D show how they can cross over and introduce new combinations of genes or alleles into the eventual daughter cells. Looking at the eventual products, you notice that when the purple chromosome separates, one of the daughter cells will have a capital letter A and a capital letter B. The other would have a capital letter A and a lowercase letter B. They're different. The blue chromatid would have a lowercase a and lowercase b, and a lowercase a and an uppercase b. So you have four different possibilities in those two alleles, A and B. If you consider that the human genome has 20 some odd thousand genes, the number of possibilities of combinations is millions. So introducing genetic variation is very easy when you have crossing over occur. It only has to occur once or twice. It doesn't have to occur very many times to have just thousands upon thousands of different combinations possible. That's really the end of this portion of meiosis, the introduction, if you will, and talking about crossing over. In the second video, we're going to talk about meiosis 1 and meiosis 2 specifically and see what occurs in each of the stages. Thank you, and bye.